My name is Daniele Baldo. I am the Communications Officer for AgroServ, and we are here for a new webinar in our series. Today, to moderate, we have Perrine Florent, who works at INRAE and is the most titled person in the AgroServ consortium, I would say, to moderate this webinar. So, Perrine, please. I leave the floor to you for the rest of the webinar. Thanks.
Thank you. Yeah, I think just a brief introduction on what AgroServ is. So that's a European research infrastructure project designed as a collaborative platform to support transdisciplinary solutions. It connects researchers, research facilities, industry stakeholders, policymakers, and farming community to accelerate agroecological research and innovation. So today's webinar focuses on SmartCow, so one of AgroServ's research infrastructures, and how it supports concrete research actions to reduce emission in livestock systems. Perfect. So I'll briefly introduce today's speakers. So first we have Cecilia Cajarville, a researcher at Synthesis Nutrition consulting company based in Barcelona in Spain. So Cecilia is a user of AgroServ Transnational and Virtual Access Program, and she will present her project enhancing sustainable beef production, the impact of anaverin on performance and methane emissions. We'll then hear from Caroline Durfee from SRUC, so Scotland Rural College, who will present the SmartCow Service Beef and Sheep Research Centre, which is involved in Cecilia's project. Finally, Frédéric de Haran from CRO, so Walloon Agricultural Research Centre, will showcase SpectraCrow, another SmartCow service available through AgroServ, illustrating the diversity of services offered within this research infrastructure. Throughout the webinar, you can submit your questions using the Q&A chat box. I'll check the 4 questions between presentations. And if we have time, we'll also have a dedicated discussion session in the end. So if you have questions, just keep asking in the chat box. Okay, with that, I'm very pleased to give the floor to our first speaker. So Cecilia, go ahead.
Perfect. Well, I'll try to tell you in brief our story about the project we presented and was funded by the AgroServ service, which is named Enhancing Sustainable Beef Production using an additive, anaverin, on performance and methane emissions by cattle. And first, as an introduction, I would say that ruminants are very efficient resource users because they basically can convert herbage in high quality animal protein. So it's very useful for humans. And in this sense, they are net protein producers. They produce more protein than they consume. And this is thanks to rumen fermentation, which also produce methane, emits methane to the environment, and this is where our problem is focused. So if we have a little look on how the greenhouse emissions are distributed in different production systems, we can see that they are quite different in ruminant production systems than in monogastric production systems. In ruminants, you can see in the graph that over 50% of the total of the emissions are methane, and this methane is produced by enteric fermentation. And why we consider methane so important? First, because it's a very potent greenhouse gas. It has a higher heat trapping than CO2, over 30 times, but also it has a shorter atmospheric lifetime. So its reduction can produce effect in a short time respect to global warming. So that's why it's so important. And how is methane distributed? Inside methane livestock production? Well, if you see this graph, more than 50% of the methane produced by ruminants is produced by non-dairy cattle, means beef cattle. That's why the beef cattle is nowadays our main target. So there are several additives that have been tried to demonstrate their effect as methane reducers. Some of them are very effective, but their adoption has been limited mainly due to safety concerns. Some are because are synthetic, they have potential toxicity, they leave potential residues, and that's why there's a strong interest in developing natural and safe and effective, of course, feed additives for the reduced methane. And here then we present the anabrin, the additive we are studying. This additive is a blend of essential oils, tannin, and bioflavonoids. And let me tell you in brief that this additive was developed by a very well-known Switzerland scientist, Ricardo Loza, and it was in first developed to reduce the transport, the stress vinculated with the transport of live animals, the immunosuppression, and to improve in general the situation of animals submitted to a long-term transport. Later on, the methane reduction properties was demonstrated. Briefly, the essential oils are from cloves, from geranium, and for coriander seeds, which are known by their antimicrobial and modulate rumen microbiome. Also contains tannins from chestnuts, which are known by the improvements in protein digestion and their anti-inflammatory properties, and also by their potential methane reduction properties, and also by olive tree bioflavonoids, known as antioxidant agents and anti-inflammatory components. So we have former reports that confirm that this product has the capacity of the potential to reduce methane, both in vitro and in vivo, and in different species, in sheep and steers, in milk cows and dairy cows, with reductions in methane that come up to the 17%, which is not bad. This product has been validated by the Carbon Trust, and also is a potential source to obtain carbon credits by farmers. In fact, in Uruguay is close to approve the product for farmers as a reducer of emissions by the verified carbon standard of Verra. And which was our need and why for this project in specific? Well, first of all, Spain is one of the top beef producers in Europe. It has the producers, the farmers, have a demand for sustainable intensification because intensive production of beef cattle is increasing in Spain. Spain has a very particular beef fattening system. It's very intensive. It's more intensive than the systems in other parts of the world. And also, if we want to certify carbon for the carbon credit programs, we need to validate the product in the context of the system where it will be used. So that's why we needed to evaluate the product on animal performance and methane emission in this type of beef intensive fattening systems. And we also had the necessity of integrate all the information that is dispersed about the product. So this is the story of the presentation of our project. We were informed that it was the opportunity of AgroServ platform, and we could present the project which finally, fortunately, was selected. And I want to say here that because some people assisting in this event are future applicants, and I have to say that it's not easy to apply to the AgroServ service because you have to choose very well the service. You need clear objectives, and you have lots of services that you can contact, and sometimes it's a bit difficult to select which is the service that provides the opportunity that you need. But if you can arrive to that, it's huge. It's very good. It's a very good experience, and the team of AgroServ helps a lot for this. The strategy we selected in accord, obviously, with the installations that accepted our proposal, was basically in two steps. The first steps are animal trials using the SmartCow infrastructure, two services of this structure. And a second step where we want to integrate data analysis, and we will use the agroecology virtual research environment of livestock infrastructure. See in the European map that we are going to work in a station of IRTA. This station is located in a beef producer farm, Agromont, Agropecuaria Mongay, which is very interesting because here we have a very close relationship with farmers, with the farmers that potentially will use our knowledge. And also we are going to work in Scotland, in the SRUC. Caroline can explain better than me, but they are in Edinburgh. And after that, we will concentrate our efforts in the livestock platform. And see in the map that we are arriving to a really interesting group of services, different services, which complement and enrich our potential work. Let me in brief tell you what we will do in each station. First, in the Agromont, in the Agropecuaria Agromont in Lleida, near here in Catalonia, with the IRTA, we will do here an experiment with an important group of animals. We will use 102 growing finishing bulls. During the experiment will last 6 months, and we will use a traditional Spanish diet. As I said, very concentrated diet, 90% concentrate, 10% straw. That's very huge concentrate. We will use 2 treatments, control and anabrin. And we will work here in a traditional performance trial, which will also aggregate individual eating behaviors, stress biomarkers. And it's interesting to comment that in a separate budget, because this platform also offers that opportunity, we will use sniffers to measure methane emissions installed by IRTA in each pen, in each of the experimental pens. And also the company, Vetos Europe, acquired a greenfield system, which is one of the systems more accepted as to measure methane emissions all over the world. And we installed at the farm, at Agromont, where we will do the experiment. So this will allow also not only to evaluate the product, the additive, but also is interesting because we will compare the technology of sniffers, which is in the future will be very useful in practical situations. It's easy to install it in a in a system, in a commercial system, and a proven methodology. This is an aggregate of the original project. In parallel, we will work with the SRUC Center in Edinburgh, and here we will use methane chambers, respiration chambers. This is very important because it's the real gold standard method to measure emissions is these methane chambers, because they measure all the emissions that the animals produce. So here we can arrive to an experiment very controlled with 12 animals in 2 treatments, crossing the treatments, and using the model of a Spanish fattening diet. Finally, we will work integrating data analysis. We will use the LifeWatch ERIC platform, which is not in the SmartCow services, but is very good to use all the trials we have. We have several experimental trials. The 2 we will do now with this project, and other ones in other situations. And also, the company has several field trials. So our aim is to use all this data to predict the efficacy of anabrine in practical situations. We will see how far we can arrive, but we think this will be a very good tool to put in different scenarios the product. In addition, and based on the opportunity of this project, Vetos Europe provided a grant to a Uruguayan PhD student, Camila Moreira, who is going to work here with us in Spain under Uruguayan Spanish supervision, which is very good to reinforce the previously existing arrangements and collaborations. So our expected project outcomes are, well, the core deliverables, or the originally deliverables, which is to prove that the product is the efficacy of the product to reduce methane emissions and improve animal productive efficiency. And I have to point out also another extended strategic value, which is the opportunity to train a new PhD expert using the direct company investment, which is good, and as we said, to advance the development of a practical methane measurement method, as is the sniffer method, which we will compare with the greenfield system. So that's it. That's all. I want to thank you for the attention, and I will try to respond any question.
Thank you, Cecilia. That was a very great presentation and a good example as well of what AgroServ can help servicing. So I don't see any question for them at the moment, but we can come back later if there is any coming after. In the meantime, so share again. So before moving forward, if I manage to find, yes, perfect. So before moving forward, I just want to give a brief introduction about SmartCow within AgroServ. So SmartCow brings together key European cattle research infrastructures of offering researchers easy access to 12 major facilities across 7 countries to support sustainable cattle production across diverse system and scientific fields. And so just to mention that beyond the services that will be presented just after here today, there's also many others that are available. And if you want to, you can check them out on the AgroServ catalog that is available on the website. And so we encourage everyone to just go and explore them. And if you need more information, you can also contact me or René Beaumont, who's the SmartCow coordinator. So yeah, so on this, I will give the floor to Caroline from SRUC, who will present this service involved in Cecilia's project. Caroline, over to you.
Perfect. Thanks very much, Perrine. Can you see my slides? Perfect. Grand.
Yes.
Certainly good morning, everybody. It's still morning here in Scotland. We're an hour behind you guys. So essentially what I'm going to cover very briefly is our research service that sits within the SmartCow infrastructure, and I'll cover how we're supporting Cecilia and the rest of the team in generating some of that data that she mentioned in those specific experiments. I also have a brief summary slide that duplicates essentially what Perrine says, but basically this kind of highlights the amount of services that exist across the AgroServ platform, but also within SmartCow. So there is a website here. I assume all of you have found that website. We can always pop it on the chat, but that website provides all the detail you need about all the different types of services that exist across all of those sort of European infrastructures. So within AgroServ, there's 11 European infrastructures with 143 different services. But if you need some help with that, you can always contact somebody here or the rest of the team to explore what those services look like. So I'll run through where I'm based. So I'm based in Scotland. So the service that we're responsible for within the SmartCow infrastructure is what we call our Beef Research Centre. So I've got beef and sheep. The team that I'm involved in is beef and sheep, but the service is focused around beef. And what I'll do is I'll walk you through some of the information about the farm, the research facilities, but also the specific facilities that we're using within Cecilia's project. So in terms of the farm resource, this is where we are. We're based just outside Edinburgh in the south of Scotland. We have roughly a total farms area of 1,000 hectares, so that equates to 2,500 acres. We are split between, as a site, we're pretty extensive. You know, we're not mountain, but we're hill. So we've got, you know, roughly 656 hectares of hill land, 152 hectares of uplands and 205 hectares of lowlands, and we are split across 5 different steadings on the same farm. Within our research service, we have roughly 400 breeding cows. So we basically calve between the spring and the autumn. Most of our calvings happen in the spring, but we also have some calving in the autumn, and we represent 3 different breed types. So on our site, we have Aberdeen Angus cross, Limousin cross, as well as what we call purebred lings. in Scotland, these are a representative hill breed. So it's nice to have those on site here as well. In terms of our breeding programme, we run a mix of artificial insemination, but also natural service. And we've started to also adopt some sex semen in our breeding herd as well, just to give us a lot of control over the progeny that we want to support the research needs that Cecilia, for example, mentioned. We also have some sheep on site, but I won't cover too much about the sheep resource. Okay, in terms of the research facilities that we're using to support Cecilia's work, I have sort of two key slides. The first is our sort of capacity around individual animal productivity and feed intake measurement. So here I show essentially a bank of feed intake recorders. These are automatic electronic feed intake recorders. That allows us to batch up the animals we need for the trial. We can then monitor animal performance, so live weight gains, but also individual animal feed intake using essentially an ear tag that is an electronic ear tag. And this picture here is exactly where that trial will happen. We will batch the animals up here into the appropriate pens. We will feed them the treatments that Cecilia mentioned, and we'll record automatic feed intake before we move these animals up into what Cecilia already mentioned is the gold standard technique for measuring methane emissions. So on site, this facility is managed by a colleague of mine called Gemma Miller. So we run 6 respiration chambers. That allows us to essentially measure individual animal emissions. So within this facility, we can measure methane, carbon dioxide, hydrogen, and oxygen. But we also monitor individual animal feed intake whilst they're in this facility, which allows us to basically equate methane to kilo dry matter intake. I've also popped in another picture here on the bottom. This is the training pens that exist within that facility. That allows us to acclimatise the animals to the building, to the pen structure, and to the management practice that exists within that building and within that facility before we measure those animals in a respiration chamber. I kind of want to point out as well, more recently, we've had some significant investment in this facility, which has allowed us to increase our capability. So alongside measuring methane, carbon dioxide, hydrogen, and oxygen, we can now also measure ammonia and nitrous oxide. We've also implemented more environmentally efficient control systems and some more modern control systems with integrated data capture. This facility has been functional since 2010, so it's a long-running facility within SRUC, and we have a lot of experience in measuring methane emissions and wider gases. And just to point out the type of work that we do within that facility, so we cover a few broad themes. Obviously at the top I mentioned animal nutrition and feed additives, which is the topic of this work we're doing with Cecilia and the wider team. But we also do a lot of work around things like breeding for low methane, high efficiency, supporting the development of proxy techniques to measure methane emissions in the field. And we're doing a lot of work in understanding the microbiome and how that relates to methane emissions, but also how the host animal genetics controls its microbiome. And now with the additional capacity in measurement, we can also explore trade-offs with ammonia and nitrous oxide. So I just want to put a picture up. It's really nice to see who the team are that support all of this research work. So we have a whole range of animal scientists. These cover all of those research themes that I've already mentioned on the previous slide. And we have an excellent team of research technicians on the bottom here that basically support that high quality experimental management, as well as the high quality data capture that we need to support that work. So very briefly, Cecilia already mentioned the trial that we're running as part of this integrated piece of work through AgroServ. But basically what we're doing here is we're running a crossover design experiment. That means that animals all get access to both treatments. So we'll have 12 beef steers, and we're using the 2 treatments. So we have a control treatment and a navrin treatment. We will spend 2 weeks training the animals in those pens with those feed intake recorders to use that equipment. We will then run an adaptation period where we'll have 6 animals allocated to each treatment. And then those animals will be batched out in groups of 6, 3 of each treatment, out to our green cow respiration chamber facility, where they'll spend a few days in the training pens to acclimatise to the environment, and then we'll spend 72 hours in that respiration chamber. The animals will then come back out, and we will swap them to the alternative treatment. So that's the crossover piece. We will then adapt them to the alternative diet treatment and repeat the process again within our green cow facility. We will then collate the data and do the sort of raw data analysis, and we will have kind of popped on here on the left side of the screen the type of data that we're capturing. So we will provide all of the categorical data around the animals and a series of numerical data, including weekly live weights that we'll capture using a calibrated weigh scale, daily feed intake, including dry matter intake, and our gas concentrations, as well as all of the respiration chamber environment data. And we will provide that to LifeWatch Eric, as Cecilia already mentioned, where we will analyse that data in collaboration with the data that's captured on the other facilities, so in INTA and any previous data that's been captured using that product. So in terms of our role, just to summarise what we do, so we'll provide access to facilities, the technical support to run the research facilities and generate the data, scientific advice in developing those protocols. We provide the animal ethics and experiments approvals, the home office licensing that's needed to deliver that type of activity. We support the experimental management, data collection, collation, as well as data QA, and the data processing, analysis, and reporting. And just as a wee reminder here, this is where you'll find information about the services. If you click on the calls and resources, it'll take you to our catalogue of services. And then I've taken a screenshot. This is from yesterday, and you can browse the different type of services that exist within the different types of infrastructures that sit within AgroServ. And I'll stop. So thank you very much for your time.
Thank you, Caroline. Yeah, so great for the AgroServ catalog. That's a great reminder, and you can also find the link in the chat. Daniel, you put it there, so thank you. Perfect. So I think we can move on to. So I don't see any question for now. I'll check again later, but for now we can move on to our last presentation. So I now give the floor to Frédéric Desharons from CRO. He's going to present SpectraCrow. So Frédéric, whenever you're ready.
Can you see my screen?
Yes, we can see it.
Thank you, Perrine. As said by Perrine, my name is Frédéric Desharons. I work at the research center called CRW, which stands for Walloon Agriculture Research Center. Today I will give you a short presentation about a service that we offer within the AgroServ project called SpectraCrow. SpectraCrow, I will explain what it is and how it could be useful for your research. Just a few words briefly to introduce the CRW. CRW is a public research institute under the government of Walloon region, which is located in the south part of Belgium. It's more than 100 years old institution spread across 3 different sites and conducts research in all areas related to agriculture and the food sector. We are a relatively small institution. It's about 450 people, including 150 scientists. And besides research, CRW also provides technical support and services to farmers and authorities. Since time is limited, I will focus on one of our 9 research units. It's the quality and authentication of product unit. This is this unit is quite unique because 4 of its 9 laboratories works with vibrational spectroscopy techniques, and this allows us to study almost all possible agricultural matrices using these methods. Alors, what is vibrational spectroscopy? Sorry. Just a small reminder, this technique uses the power of the waves. We work mainly with the wavelength shown in this red box here. You may have heard of spectroscopy in the ultraviolet region, visible region, but also the near-infrared region, mid-infrared region, or the terahertz spectroscopy. The advantage of the use of these techniques, these spectroscopic techniques, we have many advantages. Sorry. Yes. We have, for example, as an advantage, it's a little or no sample preparation. For example, we just need to have, for example, to dry or to grid, depending of the method. We don't need to have additional chemical reagents. It's fast measurement, even on moving samples. It's non-invasive methods. They do not alter the samples. It could be interesting. We have the ability to measure multiple components in a single scan, and generally they are cost effective because we save a lot of time. This method could be very interesting if you want to transfer on the field, and also to, for example, to create to use proxies to, for example, control something on the field, or to have a lot of measurements, for example, to make geneticists at large level. Our expertise, our unit has a strong expertise in this field. We started using spectroscopic methods in 1979, and since then we have acquired many instruments covering all spectral ranges. Now we have a lot of measurements in NIR, in MIR, in Raman, in near imaging. Also, we have a lot of portable devices that we call baby NIR. Also, we have camera in hyperspectral camera, but also we have apparatus for inline or online system. Almost everything are available in this domain in our unit. Of course, having instruments it's not enough. We also need to process and analyze the data, and we use a lot of chemometrics to develop our models. We have several data analysis in our team, and we also run ISO 1725. We are ISO, yes, we are accredited laboratories for reference analysis using traditional methods. We have published extensively. We have published a lot in these areas. We must be around 500 scientific publications and maybe more in this field. Because of this reputation, many instruments manufacturers come to us to test their new device and to transfer prediction models. What is SpectraCow? The idea behind SpectraCow is to allow research team to benefit from our expertise. We can help you to perform accurate analysis on your samples, to use our spectral database, or to assist you in creating your own models. Over the years, we have built large database for many types of samples, and we have also developed numerous prediction models, especially for dairy production, but not only. For example, we can predict, it's just a small list, but we have prediction for to measure the feed composition, but also the milk quality, especially the fatty acids, for example. But also we have models to follow the animal health. We had also created efficiency models. An interesting models also it's the methane emission models. We have created models to predict the eructate methane from the milk spectra, but also now we work on the feces spectra measured by IRTA to predict for the non-producing animals of milk the methane. And it seems that we have interesting results on these fields. Also, we are able to predict welfare of the animal. We do the works and much more. Practically, all of this research are published. We can produce paper on if you want. Possible services of SpectraCow could be to study the feasibility for qualitative or quantitative aspects. We can also make measurement of samples for other transnational farms. We can develop and validate a method. We can help to build and maintain spectral database. We can also we have a predicting services, and we can also standardize that it's important the NIR or MIR networks of apparatus. We have a recognized expertise in this specific domain. And also we can transfer models of your to your instruments, even if these apparatus are not from the models that we create, based on the instrument that on which we created it. In conclusion, time is short, and so I can go into details, but if you have a specific question, an idea, a compound that you want to predict, please contact me, and I will try to answer you. Many configurations are possible to integrate our service in your project. Feel free to send me an email at the address here. With your idea or question, and I will do my best to answer quickly. Thank you for your attention.
Thank you, Frédéric. That was very interesting. So, yes, do not hesitate to contact anyone if you are interested for SpectraCow. Or any other services that was presented, so SRUC or any others that are in SmartCow, we'll be happy to help if if you have an idea and you need our help for that. So we don't have question at the moment. Do have a bit of time, but so wait, I can't find my. So I'll just like to mention quickly. Can I show? Yes. If I try to find my screen again, perfect. So so I'd like to mention so that within AgroServ, we've had already 4 calls with different projects so involved in it, such as the one Cecilia just presented today. And so I'd like to highlight the fact that we're going to have our next AgroServ call for access that will open so in February 2026. So if you need any information, there will be this will be available soon on the AgroServ website. So we encourage you to keep an eye on it if you are interested at some point to apply. Perfect. So I don't know if any of you want to add anything. And if not,
Sorry.
yes. No, go ahead, please.
I would like to say that this is a very good opportunity to access to really first line research platforms. So if I could suggest someone, I would say that have clear objectives and take a time to navigate among the different resources you can use, because it's very rich. This is a very important group of services, and you can have them at the hand. That would be my advice.
That's amazing. Thank you. So yeah, so don't hesitate to check out and for our next call and the one after as well. So well, thank you a lot, Cecilia, for your words. Perfect. So I don't know if you can see my screen because I can't see it's shared. Yeah, okay, perfect. Amazing. But so yeah, just to say with that, just I'd like to thank all of our speakers with their brilliant contributions. Thank you so much. Thank you, Daniele, for organizing the webinar. And to all of you for joining, of course. We hope this AgroServ Connect webinar was very useful for you and inspiring. So yeah, have a lovely day. Bye-bye.
Bye.
Bye, Perrine.
Bye.